Usually when we talk about a medical diagnosis, there's this built-in expectation of structural precision, right? Like you break your arm, you go to the hospital, and the X-ray produces this high-contrast image.
Right, a jagged white line across a dark background.
Exactly. The doctor points at it, you look at it, and the problem is neatly defined. It's visible.
It provides this immense psychological comfort. We really crave binary systems, A bone is either broken or it's intact. An organism is a plant or an animal.
Yeah, we like the world filed away in discrete little boxes.
We do. But the reality is, the moment you step outside of those highly controlled, sterile environments, that metaphorical X-ray machine it shatters.
It completely breaks down. You realize the diagnostic landscape of the natural world is profoundly murky. And today's deep dive is going to wade straight into those muddy waters.
I mean, we are looking at a stack of sources today that span, well, it's wild. It covers fungal taxonomy, veterinary toxicology, advanced organic chemistry, and surprisingly renewable energy engineering.
Which is quite the mix. Our mission today is to take a seemingly simple walk in the woods and connect the things growing right there on the forest floor directly to the cutting edge of modern science.
Specifically, we are focusing the lens on a very specific, highly complex genus of mushrooms called Cortinarius. And we should probably establish the core paradox of this genus right from the outset.
Yeah, please do, because it's fascinating.
So this single family of fungi harbors both silent, lethal assassins that are capable of shutting down mammalian organ systems.
Which is terrifying.
Right. But at the same time, they are masterful chemical architects. They produce compounds that could literally act as the light harvesting engines to power human cities.
It's just wild. So whether you are someone who casually forages on the weekends, or maybe a dog owner constantly pulling random objects out of your pet's mouth, or, just someone tracking the transition to green technology, this deep dive is going to fundamentally shift how you view a damp forest floor.
Okay, let's unpack this. Before we can investigate the chemistry of what these mushrooms do, we have to look at the historical nightmare of figuring out what they actually are.
Oh, it really is a nightmare. We're essentially looking at a forensic cold case that spans centuries.
Right, because the visual identification of the Cortinarius genus is just notoriously difficult.
To see why, we can examine a specific group that has been the subject of fierce debate among mycologists for, well, literally hundreds of years, the Cortinarius collinitus group.
Also known as the blue-girdled webcap, right?
Exactly. And if you encounter this in the wild, it really demands your attention. The sources describe a convex cap maybe 3 to 10 centimeters across, displaying this rich orange-brown hue.
But the defining characteristic isn't the color at all, is it? No, not at all. It's the texture. In a moist environment, the entire cap is encased in a thick, gelatinous slime.
Slime.
I know, it sounds gross. Botanists refer to that slime layer as the gluten.
Gluten, okay.
And from an evolutionary perspective, that gluten serves a very specific purpose. It acts as a moisture retention barrier during dry spells, and it creates this physical defense mechanism against grazing insects.
Like they would just get bogged down in it.
Exactly. It stops them from easily chewing through the delicate fungal tissue beneath. And if you look beneath that defensive slime, you find gills that emerge pale and kind of grayish violet.
But they don't stay that color.
Right. They eventually turn a rusty brown as the mushroom reaches reproductive maturity and drops its spores.
And the blue girdled part that comes from the stem, it's this sturdy, white, cylindrical stalk, but it's wrapped in these scaly transverse bands.
Yeah, those bands are actually the remnants of a universal veil that protected the mushroom as it pushed its way up through the soil.
And in the specific species, the slimy remnants of that veil are tinted an intense lilac blue, hence blue-girdled.
Precisely. But we also cannot ignore the ecological context here. Cortinarius collinitus is an ectomycorrhizal fungus.
Which means it's not just breaking down dead leaves, right?
Right. It does not simply decompose dead matter on the forest floor. It engages in a highly complex symbiotic chemical exchange with the root systems of living trees. The mycelium, the underground network of the fungus, literally wraps around the tree roots like a glove.
So they're physically connected.
Intimately connected. The fungus acts as an external digestive system. It breaks down minerals in the soil and pipes them directly into the tree.
And what does the fungus get in return?
The tree pumps liquid carbon manufactured up in the canopy through photosynthesis straight down into the fungus.
Wow. So it is completely tethered to a specific host. The sources indicate this particular mushroom strongly prefers coniferous trees, specifically Sitka spruce, and it thrives in highly acidic, well-drained soil.
That's the profile, yeah.
So it seems like a solid checklist. Orange-brown, slimy cap, blue bands on the stem, growing under a spruce tree. I mean, it sounds like a straightforward visual diagnosis.
Which brings us to the diagnostic trap. Let me introduce the biological twin, Cortinarius mucosus.
Also known as the orange webcap, the trickster of the forest floor.
A very apt description, really. Cortinarius mucosus belongs to the exact same subgenus Mixaceum, meaning it shares that defining genetic trait of producing a viscid, glutinous outer veil.
So it also has the slime.
It does. It presents with an almost identical sticky orange-brown cap, but the subtle deviations are where the danger lies.
Okay, what are the differences?
The stem of C. mucosus is generally cream or pure white. It completely lacks any of those lilac or blue pigments in the slime bands.
Oh, so no blue girdle.
Right. Also, its microscopic spores are slightly narrower, and its host preference shifts. It actually avoids the spruce, preferring to form its mycorrhizal networks with pine trees in sandy, acidic soils.
Okay, so different stem color, different tree. There's also a detail in the research regarding Cortinarius mucosus that is genuinely unsettling.
The mercury absorption.
Yes, specimens gathered from northern Poland were analyzed, and researchers found they bioaccumulate massive concentrations of mercury from the surrounding soil.
It's incredible. The fungus is actively drawing toxic heavy metals out of the earth and storing them right in its cellular structure.
So even absent an inherent organic toxin, the mushroom basically acts as an environmental sponge for poison.
It does. But the broader issue here is that differentiating a blue-banded stem from a white-banded stem in a pristine, well-lit laboratory setting is easy.
Sure. Under a microscope.
Right. But differentiating them in a dark, muddy forest after 3 days of heavy rain, when the slime has completely washed away and the colors have faded to gray, it's nearly impossible.
Yeah, I can imagine.
And this visual ambiguity isn't just a hurdle for amateur foragers, you know? It completely derailed the scientific classification of these organisms for 2 entire centuries.
Because the foundational texts of mycology were essentially operating on faulty data.
Exactly.
The original botanical documentation of the blue-girdled webcap back when it was classified as Agaricus collinitus was published by James Sowerby in 1797.
And we have to consider the reality of 18th century botany. Sowerby was drawing these specimens by hand.
Just sketching them in a notebook.
Right. And modern forensic taxonomy, when scientists look back at his original plates, strongly suggests Sowerby was actually illustrating a completely different, unrelated species.
Oh, wow.
Yet his drawing became the anchor point for the species' identity. Then, by 1821, Samuel Frederick Gray moved the name into the genus Cortinarius, but Gray's written description highlighted scales on the stem that actually belonged to yet another distinct species, Cortinarius trivialis.
It's like a taxonomic disaster. Then Elias Fries, who's basically one of the founding fathers of modern mycology, included it in his major 1821 work, Systema Mycologicum. But Fries blended features from multiple different mushrooms into a single, highly ambiguous description.
It was chaos. The scientific community was playing a multi-generational game of telephone. They were basing new research on flawed descriptions of misidentified drawings.
Wait, so relying on an old field guide to identify a mushroom is basically like using a 19th century pirate map to navigate the modern New York subway system.
That's a remarkably accurate analogy. The shapes might look vaguely similar, but the underlying infrastructure is completely different.
Yeah, the stakes are incredibly high. If a field guide from, say, the 1980s lumps 5 different regional mushrooms under a single European name and assumes they are all safe to eat simply because they share a similar cap shape, the reader is walking into a physiological minefield.
They absolutely are. The assumption of global uniformity was the primary failure here. Mycologists historically believed that if a mushroom in Massachusetts looked morphologically identical to a mushroom in a French forest, they must be the same species.
But they aren't.
No. We now know that ectomycorrhizal fungi, because of their intimate evolutionary ties to highly specific local trees and local soil chemistries, are intensely geographically isolated.
Which forces a total shift in methodology. I mean, if we can't trust the visual shape of the mushroom and we can't trust the historical texts, we have to look at the structural code of the organism itself. We have to move to DNA barcoding.
Exactly. Fungal taxonomy is currently undergoing this massive, necessary purge. Scientists are no longer relying on the color of a spore print on a piece of paper. They are extracting genomic DNA directly from the gills of the mushroom.
And the primary target for this sequencing is the internal transcribe spacer, or the ITS locus, right?
That's one.
My understanding of the ITS region is that it sits between the genes that code for ribosomal RNA. And because the spacer itself doesn't actively code for the critical structure of the ribosome, it isn't subjected to the same intense evolutionary pressure to remain identical over millions of years.
You've got it.
So it accumulates mutations much faster than the functional genes around it.
That is a brilliant way to conceptualize it. If you look at a highly conserved critical gene, a mushroom from North America and a mushroom from Europe might look identical, simply because any mutation in that critical gene would have killed the organism.
Right, the organism wouldn't survive the mutation.
Exactly. But the ITS region acts like an evolutionary scratch pad. The harmless mutations accumulate rapidly over time, creating a highly specific genetic barcode that clearly distinguishes a species in a New England oak forest from its cousin in a Bavarian pine forest.
The sources also note the utilization of the 28S locus, specifically the 5' end of the large subunit ribosomal DNA.
Yes, as a secondary confirmation.
By running these specific genetic sequences through polymerase chain reaction to amplify the DNA, and then mapping them with phylogenetic software, scientists are finally tearing down those umbrella classifications.
They are proving that these so-called species complexes are actually comprised of dozens of distinct, highly endemic organisms.
And untangling this taxonomy leads us directly to the clinical consequences. Because when you strip away the visual confusion and analyze the true chemical reality of these geographically distinct fungi, you uncover the silent assassins lurking in the genus.
You do. It's a sobering reality.
Let's transition from the abstract world of taxonomy to a highly specific, very physical reality. We are looking at a landmark 2016 study published in the journal Toxicon. This research pivots from identifying the mushroom to understanding the terrifying cascade of physiological events that occur when a mammal actually consumes one.
The focus of this particular study is Cortinarius armillatus. The research team was conducting a routine survey of mutually beneficial root symbionts in the Harvard forest.
Which is a heavily monitored ecological research site in Petersham, Massachusetts, right?
Right. They were harvesting these specimens from moist, acidic soils beneath the canopies of red oak and paper birch trees.
And from a visual standpoint, Cortinarius armillatus is the definition of unassuming. It is a dull, brownish-orange mushroom. It features a whitish ring on the stem called an annulus, which catches the rust-colored spores as they fall from the gills.
It looks like 1,000 other generic brown mushrooms on the forest floor. You would walk right past it.
But this team didn't just photograph it. They sequenced the ITS barcode to confirm its identity against the neotype specimen, and then they subjected the tissue to rigorous chemical diagnostics.
And the results of that chemical assay fundamentally altered the toxicological map of North America. They definitively proved that Cortinarius armillatus, a native North American species, biosynthesizes a compound called Orellanine.
Which is a huge deal.
Massive. Prior to the sequencing, the prevailing medical consensus was that Orellanine was largely restricted to a handful of lethal European species, specifically Cortinarius orellanus and Cortinarius rubellus.
So the immediate danger isn't just across the Atlantic. It is growing alongside the hiking trails in Massachusetts.
Exactly.
To grasp why discovering Orellanine locally sent such shockwaves through the toxicology community, we have to look at the structural architecture of the molecule itself. The literature defines Orellanine as a tetrahydroxylated diane-oxidized bipyridine.
That's quite a mouthful.
It really is.
Yeah.
Can we break that intimidating nomenclature down into like a functional blueprint?
Sure. Imagine two hexagonal rings consisting of carbon and nitrogen atoms. See, these are called pyridine rings.
Okay, 2 rings.
Right. In bipyridine means two of these rings are linked together by a central bond, forming a rigid, flat core. Then stud that rigid core with hydroxyl groups, which is an oxygen bound to a hydrogen and specific nitrogen oxide bonds.
The functional shape of that molecule is what matters, right? Because when I looked at bipyridine structure in the sources, it bears a striking, unsettling resemblance to synthetic commercial herbicides.
It does.
So chemically, this natural mushroom toxin looks incredibly similar to paraquat and diquat commercial synthetic weed killers.
The structural homology is undeniable, and the mechanism of toxicity shares certain parallels. It also resembles MPTP, which is a synthetic neurotoxin that selectively targets and destroys dopaminergic neurons in the brain, inducing permanent Parkinsonian symptoms practically overnight.
Wow. But Orellanine doesn't target the brain, does it?
No. Orellanine is a profoundly potent nephrotoxin. It bypasses the brain and the liver almost entirely.
Just floats right past them.
Exactly. It travels through the bloodstream until it reaches the kidneys, where its specific molecular geometry acts like a master key. It slips perfectly into the receptors of the renal epithelial cells.
Once it breaches those renal cells, how is it actually dismantling the kidney? Because it isn't an immediate, violent reaction. If someone accidentally eats a highly toxic mushroom like the death cap, they're generally violently ill within hours, which prompts emergency medical intervention.
Orellanine operates on a completely different timeline.
It employs a biochemical stealth attack. Once inside the kidney cells, orellanine begins generating massive amounts of reactive oxygen species.
Free radical. Right, free radicals. These free radicals begin aggressively stealing electrons from the cell's structural proteins and its DNA. The cell tries to defend itself by deploying antioxidants like glutathione, but it's not enough.
No, because the orellanine continually generates more radicals, eventually completely depleting the cell's defenses.
So the kidney is slowly burning from the inside out at a cellular level.
It is a slow, relentless oxidative fire. The initial consumption of the mushroom might produce only mild gastrointestinal discomfort, or honestly, absolutely zero symptoms at all.
Nothing.
Nothing. The catastrophic cascading failure of the renal tissue might not manifest clinically until 7 days or even up to 14 days after that single exposure.
A 2-week delayed fuse. That is horrifying. If a patient presents at an emergency room with sudden acute renal failure on a Thursday, no doctor is going to think to ask, hey, did you happen to eat a brownish-orange wild mushroom while camping the weekend before last? Never. The massive delay completely severs the causal link in the mind of the patient and the physician. It pulls the diagnosis deep into those muddy waters we talked about.
And the clinical implications of that delayed onset are particularly dire when we look beyond human exposure and consider veterinary medicine. The Toxicon study highlights a jarring discrepancy. Prior to this research, there was not a single confirmed, documented case of Cortinarius poisoning in a dog or cat in North America.
Which is statistically impossible. The researchers do not believe this is because animals aren't eating the mushrooms. They believe it is a massive failure of diagnostic infrastructure.
Because dogs are indiscriminate eaters. They explore the world with their mouths. They will bolt through a backyard, find a patch of raw fungi under an oak tree, and consume it before the owner even registers what happened.
Veterinarians are very well trained to look for hepatotoxicity, rapid, severe liver failure caused by the amatoxins found in the Amanita genus. If a dog eats a death cap, the symptoms are severe and immediate.
Right, they know what to look for.
But if a dog ate a Cortinarius mushroom, the delayed kidney failure 2 weeks later would likely be misdiagnosed as an idiopathic infection or simply age-related organ decline.
And more critically, veterinary diagnostic laboratories in North America literally lack the analytical protocols to test a blood or tissue sample for orellanine.
You cannot diagnose an invisible toxin if you do not possess the machinery to see it.
Exactly. And that diagnostic void is what drove the research team at Iowa State University to engineer 2 entirely new, highly sophisticated analytical tests to hunt for this specific molecule.
They developed an HPLC protocol, which stands for high-performance liquid chromatography, and a vastly more powerful LC-MS protocol, liquid chromatography tandem mass spectrometry.
And the physical extraction process required to pull this molecule out of the raw mushroom tissue before they even put it in the machine. It's an exercise in extreme chemical precision.
Oh, it's intense. They do not simply mash the mushroom and put it on a microscope slide. They take a microscopic sample, just 15 milligrams of dried mushroom tissue.
That's practically dust.
It is. And they submerge it in 3 molar hydrochloric acid.
Wait, that is an incredibly aggressive acid. Why do they need something that harsh?
Because the orellanine is bound up tightly within the structural glycosidic bonds and complex proteins of the fungal cells. The hydrochloric acid systematically breaks those bonds, lysing the cells open and freeing the toxin into the liquid suspension.
And they have to stir it, right?
Yes, they maintain this acidic bath for 3 hours under constant magnetic stirring to ensure complete cellular degradation.
And they also have to conduct this entire violent chemical extraction in the dark. The sources note that orellanine undergoes rapid photodecomposition.
Right. If the ambient light of the laboratory hits the extracted molecule, the UV radiation will shatter the delicate bipyridine rings, completely destroying the evidence.
So every vial and test tube has to be tightly wrapped in aluminum foil.
Exactly. After the acid bath, they utilize a centrifuge, spinning the slurry at 3,000 RPM for 30 minutes.
To separate it out.
Right. The centripetal force drives all the heavy, degraded cellular debris to the bottom of the tube, leaving a clear, liquid supernatant at the top containing the dissolved toxin.
But that liquid is still highly acidic.
Very. They have to carefully titrate it with ammonium hydroxide to neutralize the acid, then run it through a series of solid-phase extraction columns, washing it with water and methanol adjusted to a highly specific pH of 1.5 using formic acid.
The exactness of that pH is vital, I assume, because at pH 1.5, the orellanine molecule holds a specific electrical charge that allows it to grip the filter column, while all the other random fungal proteins just wash straight through.
That's exactly how it works. Finally, they wash the purified toxin off the column and concentrate it down using a gentle stream of nitrogen gas.
Once they have that highly purified, concentrated extract, they inject it into the analytical machines. Let's talk about the first one. The HPLC method utilizes a photodiode array UV visible detector.
It shines a specific spectrum of light through the liquid and measures exactly how the molecules absorb that light. The HPLC protocol proved capable of detecting orellanine down to 17 micrograms per gram of mushroom tissue.
Which is impressive. But the LC-MS machine operates on an entirely different plane of physics.
It really does.
Rather than just looking at how the molecule absorbs light, a tandem mass spectrometer essentially weighs the shattered pieces of the molecule to confirm its identity.
The mechanics of LC-MS are just breathtaking. The purified liquid is pushed through an electrospray ionization chamber where a high voltage shears the liquid into a fine mist of electrically charged ions.
Okay, making a mist.
These charged molecules then fly into a vacuum chamber containing 3 separate quadrupole magnets.
So it's basically a molecular wind tunnel.
A wind tunnel that filters by atomic mass. The first set of magnets, the first quadrupole, is tuned to only allow the exact atomic weight of the intact orellanine molecule to pass through.
So everything else just crashes.
Everything else crashes into the walls and is discarded. The surviving orellanine molecules then enter the second quadrupole, which acts as a collision chamber.
They smash them.
They are bombarded with argon gas, physically shattering the orellanine molecule into smaller, predictable fragments, known as daughter ions.
And the third quadrupole filters those shattered fragments, weighing them to prove that the parent molecule was definitively orellanine, and not just some other random chemical that happened to share the same initial weight.
Exactly. And because of this incredibly rigorous multi-stage filtration, the LC-MS is roughly 2,000 times more sensitive than the HPLC. It can detect the toxin at concentrations of 30 nanograms per gram.
It is the analytical equivalent of finding a single specific grain of sand on a vast beach and confirming its exact atomic composition.
It's a marvel. And when they ran the Cortinarius armillatus samples through this newly engineered gauntlet, they found the toxin, but at an intriguing concentration. The native North American mushroom contained roughly 145 micrograms of orellanine per gram of dry tissue.
To understand the implications of that number, we have to look at the control sample. The researchers ran a parallel test on Cortinarius rubellus, the notoriously lethal European species.
And the difference is staggering.
That specimen contained an astonishing 24,000 micrograms per gram. It was literally 2.4% pure toxin by weight. So the North American armillatus contains less than 1% of the orellanine found in its deadly European cousin.
Which naturally invites the question for you listening. If the concentration is that low, is it actually a threat to you or your dog?
Right, because less than 1% seems negligible.
The toxicological reality is that a healthy adult human or a large, perfectly healthy dog might consume a single Cortinarius armillatus mushroom and never experience the cascade into acute renal failure. The kidneys might process that low dose of free radicals and recover.
But medical risk is rarely calculated based on the healthiest possible individual. If you take a senior dog who already has a subtle, undiagnosed kidney infection, or an older human forager with mild hypertension causing underlying renal stress, that low dose of orellanine ceases to be harmless.
It becomes the biochemical tipping point. The depletion of glutathione caused by that low dose pushes an already struggling organ over the edge into irreversible end-stage renal disease.
The true victory of the ToxCon study is that the diagnostic void has been filled. Veterinary clinics across the continent now have a protocol. If a dog falls ill with delayed kidney failure, the clinic can overnight a sample of stomach contents or a mushroom pulled from the yard to a diagnostic lab, and the LC-MS machine will provide a definitive, quantitative answer.
It's a huge leap forward for toxicology.
Here is where it gets really interesting. The scientific community built this incredibly sophisticated diagnostic infrastructure, the DNA barcoding, the liquid chromatography, the tandem mass spectrometry, specifically to hunt for these lethal molecules. But as they aimed these powerful tools at the fungal kingdom, they didn't just find poisons.
No, they didn't. They illuminated a vast hidden pharmacy of biochemical compounds.
They discovered that the exact same evolutionary pressure that forces a fungus to build a deadly bipyridine also forces it to build life-saving medicines.
We shift our focus entirely now to another major class of secondary metabolites produced by fungi, the anthraquinones.
We have a massive comprehensive review from the journal MDPI that catalogs the dizzying array of biological functions these anthraquinones perform. But before we look at what they do, we need to understand how the fungus actually builds them. The literature refers to the acetate route.
Right. Well, first, a secondary metabolite is a compound that an organism produces, which isn't strictly required for fundamental growth or reproduction. The fungus won't die immediately if it stops producing it, but the compound provides a massive competitive advantage.
Okay, so it's like a biological upgrade.
Exactly. Fungi build anthraquinones using a polyketide synthase pathway.
Think of it as a biological assembly line snapping together molecular Lego bricks.
That's a great visual. The fungus strings together a polyketide chain containing 8 2-carbon units. Imagine a long, floppy string of carbon atoms.
Just waving around.
Yeah. Through a sequence of highly orchestrated folding events and aldol-type condensation reactions, the enzymes within the fungus force that floppy chain to snap together into a rigid, flat, 3-ringed carbon skeleton.
The rigidity of that 3-ring structure is the foundation of everything that follows, right?
Absolutely. Once the fungus has constructed that robust, flat chassis, it can begin bolting on different functional groups, adding a hydroxyl group here, a methyl group there, an amino group on the side.
Just accessorizing the skeleton.
By making these tiny chemical modifications, the fungus creates hundreds of distinct anthraquinone variants.
And we're going to trace how these tiny structural modifications allow these molecules to interact with a staggering variety of human pathogens. The MDPI review reads like a catalog of microscopic warfare. Let's look at the genus Alternaria. Researchers isolated a specific Alternaria species living inside a soft coral in the South China Sea.
A fungus living inside a marine animal. The environmental pressure there must be intense.
I can't even imagine.
The environment demands extreme chemical defenses. This specific marine fungus produces a modified hydroanthraquinone called a tetrahydroaltersolanol.
Say that 5 times fast.
I won't try. But when researchers tested these specific compounds, they discovered they possessed profound antiviral capabilities. They specifically disrupted the replication cycle of the porcine reproductive and respiratory syndrome virus, or PRRSV.
Which is a devastating agricultural pathogen.
It is. Furthermore, related compounds synthesized by the same fungus demonstrated severe cytotoxicity against human prostate and colon cancer cell lines in a laboratory setting.
It's actively dismantling the replication machinery of a virus and inducing cell death in human tumors. It's incredible. Let's pivot to an entirely different environment. Consider the fungus Angiodontium album. This organism thrives in remarkably harsh, nutrient-poor terrestrial environments. It grows on damp indoor paper, on painted drywall, and on decaying textile.
To survive on a painted wall, you have to aggressively defend your territory against bacterial colonization. And Angiodontium album manufactures a unique class of anthraquinones named angiodontichones.
And the target of these molecules is astonishing. In clinical assays, Angiodontichone has demonstrated potent inhibitory action against MRSA methicillin-resistant Staphylococcus aureus.
MRSA is the nightmare scenario for modern hospitals. It is a bacterium that has evolved a brutally thick peptidocking cell wall and cellular pumps that actively eject standard antibiotics. We struggle to kill it with our most advanced synthetic drugs.
Yet the anthraquinones produced by a fungus growing on damp drywall inhibited the growth of MRSA at a rate 10 times stronger than chloramphenicol.
And chloramphenicol is a heavy-duty, broad-spectrum commercial antibiotic. The rigid, flat structure of the anthraquinone likely allows it to intercalate or physically wedge itself into the bacterial membrane, disrupting the structural integrity that makes MRSA so resilient.
So the shape of the molecule is the weapon. The chemical adaptations only get stranger. We have an endophytic fungus called Fusarium solani. Endophytic means the entire fungal organism lives symbiotically inside the tissues of a living plant, in this case, the Cassiel alata shrub in Bangladesh.
This endophyte synthesizes an aza-anthraquinone called bostriquatin.
And when researchers extracted bostricoidin, they utilized a fascinating preliminary screening method known as the brine shrimp lethality bioassay.
Oh, I love this test. They introduced the crude fungal extract to microscopic brine shrimp. It provides a rapid, observable metric of general bioactive toxicity.
If the extract kills the shrimp, the chemists know there is a potent molecule hidden inside.
Right, and they begin the painstaking work of isolating it.
The bostricoidin they isolated proved to be a highly effective antimicrobial and anti-cancer agent. But if we are looking for the absolute pinnacle of fungal chemical warfare, we have to look at the genus Torubiella. This is an entomopathogenic fungus.
It does not live quietly inside a leaf or on a painted wall. It actively hunts. It parasitizes spiders, scale insects, and leafhoppers.
It literally consumes the insect from the inside out.
It's brutal. To manage the immense immune response of its prey and to fend off opportunistic bacteria that might try to steal the insect corpse, Torubiella synthesizes dimeric anthraquinones.
Dimeric meaning double.
Exactly. The fungus essentially takes two standard three-ring anthraquinone skeletons and bonds them together into a massive, complex molecule called a torubiellin.
And the biological activity of torubiellin is absurdly broad. It demonstrates strong antibacterial action against Bacillus cereus, it fights off competing fungal infections like Candida, and it aggressively attacks cancer cell lines. But the standout metric is its anti-malarial capability.
Yes. Torubiellin exhibits incredibly strong toxicity against Plasmodium falciparum.
Plasmodium falciparum is the protozoan parasite responsible for the most lethal form of human malaria. It hides inside human red blood cells, rapidly replicating and destroying the cell.
And the torubiellin molecule is somehow capable of breaching the red blood cell, navigating the internal environment, and dismantling the parasite's biochemical machinery.
I have to pause and frame a broader biological question here. We are looking at a fungus that eats spiders in the jungle, and it just happens to manufacture a molecule that cures the deadliest form of human malaria. We have a fungus living inside a shrub in Bangladesh, putting immense metabolic energy into synthesizing a compound that kills human breast cancer.
Yeah.
What is the evolutionary logic here? The fungus obviously doesn't care about human oncology.
You are hitting on the central truth of natural product chemistry. Fungi are not altruistic. A fungus is a relatively fragile, soft-tissued organism trying to survive in a microscopic war zone.
Right.
Every resource, every square millimeter of a decaying log, is violently contested by billions of aggressive bacteria grazing nematodes and viral phages. The fungi survive by deploying chemical weapons.
So an anthraquinone isn't designed to cure malaria. It is designed to paralyze a nematode or rupture the cell wall of a competing bacterium.
Precisely. We are observing the result of an evolutionary arms race that has raged without pause for hundreds of millions of years. The fungi are the most prolific combinatorial chemists on the planet. As bacteria mutate to resist a toxin, the fungus mutates the anthraquinone, perhaps shifting a hydroxyl group 1 carbon over to bypass the new defense.
And we as humans are simply the beneficiaries of this ancient war.
We are, because the fundamental biochemistry of a bacterial cell membrane, or the replication enzyme of a virus, shares ancient evolutionary roots with the pathogens that infect humans. We can hijack the fungi's terrestrial defense mechanisms and stock our own pharmacies.
The functional overlap is incredible. There's one final medical example from the sources that perfectly illustrates how precise these molecular weapons have become. We look at Eurotium rubrum, a fungus found living on salt-tolerant wild plants near the ocean. It synthesizes an anthraquinone called rubramol.
Rubramol has an incredibly specific mechanism of action. It targets topoisomerase of farn. This is a critical enzyme responsible for managing the physical tension of DNA.
Which is important because DNA gets twisted up, right?
Exactly. When a cell needs to read its genetic code, the tightly wound DNA double helix has to be unwound. Topoisomerase cuts one DNA strand, allows it to relax, and then seals it back together.
If you disable that enzyme, the DNA supercoils, the tension shatters the genetic structure, and the cell undergoes apoptosis, it dies. It's a very common mechanism for aggressive chemotherapy drugs.
But rubramol does something extraordinary. In clinical tests, it physically interacted with topoisomerase, successfully relaxing supercoiled DNA. However, it accomplished this manipulation without exhibiting any cytotoxic activity against normal, healthy human cell lines.
That's amazing.
It manipulated the physical geometry of the DNA strand without triggering the catastrophic death of the healthy cell.
It is operating with microscopic surgical precision. And this brings us to a conceptual bridge. The exact same structural feature that makes these molecules so effective in a medical context, that rigid, flat, three-ring carbon chassis, also grants them a completely different set of physical properties.
Yes, the rigid framework of alternating single and double carbon bonds doesn't just allow the molecule to slip between the base pairs of DNA. That specific arrangement of electrons interacts violently with electromagnetic radiation.
Meaning it interacts with light.
It absorbs and reflects light with immense efficiency. These microscopic weapons of war happen to be some of the most vibrant, stable pigments in the natural world.
Which takes our deep dive out of the pharmacology lab and thrusts it straight into materials science and the history of industrial engineering. Because the history of anthraquinones isn't just about medicine, it is the history of color. The sources point out that the very first industrial application of an anthraquinone compound occurred way back in 1869 with a dye called alizarin.
Before the late 19th century, achieving a truly color-fast red dye for textiles was a wildly expensive and labor-intensive process. Alizarin was a natural anthraquinone extracted from the roots of the madder plant Rubia tinctorum.
And when chemists finally deduced its chemical structure and figured out how to synthesize it from coal tar, it utterly revolutionized the global textile industry. It democratized color.
And once industrial chemists possessed the blueprint for that three-ring anthraquinone core, they started accessorizing it, just like the fungi do. By bolting a nitro group onto the chassis or adding a sulfonic acid chain, they engineered a massive spectrum of brilliant blues, vibrant greens, and sharp yellows.
Colors that could permanently bond to cotton, wool, and eventually synthetic polyesters.
But that chemical revolution came with a catastrophic environmental debt. The modern synthetic dye industry relies heavily on petrochemicals and heavy metals. The wastewater generated by these massive dyeing facilities is phenomenally toxic.
Because the rigid structure that makes the dye so stable and color-fast on a t-shirt also makes it nearly impossible for the environment to break down.
Right. The research papers detail the extreme measures required to treat this industrial sludge. Facilities have to deploy advanced oxidation processes, blasting the water with ozone and ultraviolet light, just to shatter the carbon bonds of the synthetic dyes.
They even utilize bioluminescent bacteria to test the toxicity of the water, measuring how much the pollution dims the bacteria's natural glow. It is a massive ecological crisis.
And this is where the fungal kingdom re-enters the narrative, not as a poison, but as a sustainable salvation. Certain fungi, specifically the mushrooms belonging to the Cortinarius subgenus Dermacybe, offer a naturally occurring, highly stable, and fundamentally biodegradable alternative to synthetic petrochemical dyes.
Because these mushrooms naturally biosynthesize massive quantities of pigmented anthraquinones. The literature highlights specific molecular variants like emodin, dermacybin, and dermarubin.
Historically, indigenous populations and traditional weavers in Nordic countries have utilized these specific Dermacybe mushrooms for centuries.
To dye their clothes.
By boiling the fungal tissue, they extract these pigments to dye wool, yielding remarkably rich, color-fast yellows, burnt oranges, and deep blood-red hues. The pigment bonds tenaciously to the protein fibers of the wool, yet it remains ecologically benign.
But modern material scientists are no longer just looking at these fungal pigments to sustainably dye winter sweaters. They're looking at the exact physical properties of these molecules to solve the global energy crisis.
This is truly cutting edge.
This brings us to the most radical concept in the ResearchGate papers. Engineers are actively harvesting these specific mushroom dyes, the emodin and the dermacybin, to construct dye-sensitized solar cells, or DSSCs.
The premise sounds like science fiction generating electricity from mushroom dye, but the underlying physics are elegantly simple. A dye-sensitized solar cell is essentially an artificial leaf. It is a mechanical attempt to mimic the biological process of plant photosynthesis.
We need to break down the anatomy of a DSSC to understand how the fungus fits into the machine. A standard solar cell in this category relies on a semiconductor film, typically constructed from a microscopic layer of titanium dioxide, or TiO2.
Titanium dioxide is an excellent semiconductor, but it has a massive functional flaw when it comes to solar energy. Its band gap, the amount of energy required to knock an electron loose, is very wide.
So it only absorbs high-energy light.
Exactly. It only absorbs high-energy ultraviolet light. It is completely transparent to the vast majority of the visible light spectrum that pours out of the sun. It needs a chemical adapter plug to capture that visible light.
It needs a sensitizer, needs a dye. And this is where the Cortinarius extract comes in.
The engineers take a crude chemical extract from the Cortinarius mushrooms, a liquid teeming with those brightly colored emodin and dermacybin anthraquinones. They submerge the porous titanium dioxide film into this fungal bath.
And this is where the specific accessory groups on the anthracnone chassis become critical. The research highlights the hydroxyl groups, the oxygen and hydrogen pairings, and the carboxyl groups extending off the main carbon rings.
Those groups act as microscopic chemical grappling hooks. They allow the fungal pigment molecule to physically bond to the surface lattice of the titanium dioxide. The dye perfectly coats the semiconductor.
So the adapter plug is connected. What is the physical mechanism that occurs the moment sunlight hits that coated surface?
The fungal anthraquinones are evolutionary masterworks at absorbing visible light. When a photon from the sun strikes the rigid, conjugated double bonds of the dye molecule, the energy is absorbed. This energy violently excites an electron within the dye, bumping it up to a higher energy state, what chemists call the lowest unoccupied molecular orbital.
LMO. And because the dye is physically anchored to the semiconductor, the excited electron doesn't just fall back down and release heat, it cascades directly into the conduction band of the titanium dioxide.
Exactly. The titanium dioxide channels that flowing electron into a conductive circuit. The moving electrons provide electrical power to an external device, and then they flow back into the cell, where an iodine-based liquid electrolyte acts as a chemical shuttle, physically carrying an electron back to the original dye molecule to reset the system.
The mushroom pigment acts as a relentless, photon-powered engine, constantly absorbing light and firing electrons into the grid. The elegance of the system is undeniable.
It's a beautiful piece of engineering.
But we have to look at the hard data provided in the papers. A prototype DSSC, utilizing the crude, unpurified extract from the Cortinarius fungi, achieved a photoconversion efficiency of 564%. The engineers documented an open-circuit voltage of 541 millivolts and a fill factor of roughly 66%. I have to play the skeptic here.
I knew you would.
An efficiency of 0.64%. The rigid silicon solar panels bolted to the roofs of modern homes operated at efficiencies between 15% and 20%. Why should the energy sector care about a fungal solar cell that operates at less than 1% efficiency?
That skepticism is the standard gauntlet for any nascent technology. But evaluating a DSSC solely against the peak efficiency of a silicon panel ignores the broader ecological and economic context.
Okay, how so?
Standard silicon photovoltaics are highly efficient, but their creation is violently destructive. They require the aggressive mining of rare earth metals and metallurgical-grade silicon.
The manufacturing process requires heating massive quartz crucibles to thousands of degrees, consuming massive amounts of grid power, and utilizing highly toxic solvents like hydrofluoric acid. Even if the energy they eventually produce is clean, the physical panels are born from ecological trauma, and they are notoriously difficult to recycle at the end of their lifespan.
Precisely. Fungal DSSCs belong to the third generation of solar technology. Their value proposition is not peak efficiency. It is radical accessibility and sustainability. They can be manufactured at room temperature. They do not require vacuum chambers or toxic solvents.
Just cheap glass and mushroom dye.
The primary components are cheap glass, non-toxic titanium dioxide, and a biodegradable dye extracted from naturally abundant fungi or agricultural waste.
Furthermore, the literature points out a functional advantage. While silicon panels suffer massive efficiency drops in low-light conditions, or when partially shaded, dye-sensitized solar cells operate remarkably well under diffuse light. They can generate power on heavily overcast days or even indoors under artificial LED lighting.
They are not designed to replace the massive solar farms powering industrial grids. They are designed to power the millions of low-drain devices entering the market, indoor environmental sensors, Internet of Things electronics, and wearable technology.
And more importantly, the sources explicitly connect this fungal technology to the United Nations Sustainable Development Goals, specifically the mandates for zero hunger and affordable clean energy.
The implication being that developing nations, which might lack the billions of dollars required to build semiconductor foundries, could literally grow their own solar infrastructure.
They could cultivate vats of natural fungi, extract the dyes using simple chemistry, and manufacture cheap, functional solar cells to provide reliable, off-grid power to remote medical clinics or agricultural centers.
Exactly. And that 0.64% efficiency is merely the starting line. As organic chemists learn to purify the extracts and tweak the carboxyl anchoring groups to bind more tightly to the titanium, that efficiency curve will inevitably rise.
The potential is staggering. We have traced an incredible narrative arc today. We began by examining the Cortinarius genus, a group of mushrooms so morphologically deceptive that it forced the entire field of mycology to abandon visual identification and rely on molecular DNA barcoding just to comprehend the biodiversity of a single forest floor.
And then we explored the dark side of that chemistry, examining how a native North American mushroom biosynthesizes orellanine, a molecule perfectly shaped to act as a stealth weapon, initiating a delayed catastrophic oxidative fire within mammalian kidneys, a threat so hidden it required the invention of tandem mass spectrometry protocols just to diagnose it in our pets.
We then witnessed how the exact same evolutionary pressure that forged those toxins also engineered the anthraquinone's rigid, versatile molecules capable of physically tearing apart the armor of MRSA, dismantling the malaria parasite inside a red blood cell, and selectively relaxing the tension of human DNA without harming the cell.
And finally, we saw how the optical properties of those identical anthraquinones could be harnessed to mimic the leaf of a tree, capturing the energy of the sun to drive a biodegradable electrical circuit.
It brings us full circle to the broken X-ray machine we discussed in the beginning. The natural world refuses to be simple. It is messy, it is dangerous, and it is infinitely more complex than a picture in a field guide.
I want to leave you with one final conceptual leap, expanding on the synthesis of everything we've covered today. Currently, to obtain these miraculous compounds, whether extracting turbilin for an experimental malaria trial or dermacybin for a prototype solar cell, we have to physically harvest the wild organism.
Which isn't sustainable on a massive global scale.
Right. But as we discussed during the taxonomy segment, geneticists have mapped the ITS barcodes, and they are now mapping the entire genomic architecture of these fungi. They know exactly which gene clusters govern the polykide synthase pathways.
They possess the source code.
The exact biological software. Consider the near future of synthetic biology. We can isolate the specific genetic sequence from a Cortinarius mushroom that codes for the production of the amodin pigment. We can utilize CRISPR technology to splice that genetic code directly into the genome of Saccharomyces cerevisiae, ordinary single-celled baker's yeast.
We already do this with human insulin. We programmed yeast to brew medicine decades ago.
We can program massive industrial fermentation vats of yeast to rapidly consume simple sugars and continuously excrete pure synthetic Cortinarius anthraquinones.
Wow.
We could literally brew the raw materials for biodegradable solar panels or highly targeted personalized cancer therapeutics with the exact same scalable, inexpensive infrastructure we currently use to brew a pale ale.
The ultimate value of the forest isn't the physical mushroom decomposing the pine needles. The true value is the chemical software running inside its cells, waiting to be understood.
Exactly.
Brewing solar panels in a vat of yeast. That is an image that fundamentally alters how I look at industrial manufacturing. Thank you for joining us on this incredibly deep dive into the dangerous, healing, and endlessly surprising world of the Cortinarius fungi. The next time you are walking through a damp forest and you spot a strange, slimy orange mushroom pushing its way up through the soil, maybe pause and give it a nod of respect, because beneath that muddy exterior, it might just be running the code to the future.